Heritable retinal degeneration happens because of mutations in key retinal structure genes. This can present as many different kinds of vision impairment, such as progressive blindness or difficulty seeing specific types of colors. Can we fix it? Sort of. We can fix it with gene therapy, theoretically, delivering a new gene using a virus, but there's really a limited region of therapeutic effects in the retina because the virus, when you inject it into the retina, doesn't really spread to infect too many cells.
This brings us to our research question: can we engineer a virus to effectively treat retinal degeneration? The answer to that is through synthetic virology, which is gene delivery with enhanced targeting abilities with resistance to immune responses. Essentially, therapeutic transgenes are swapped with viral genes via genetic recombinating techniques. Pseudotyping is used, which is optimizing viral capsid proteins on the outside for enhanced gene delivery. This is what essentially allows the gene therapy to advance beyond the injection site. Adenoviruses are commonly used because they are non-pathogenic to humans, have large transgene capacity, and have efficiency in transduction into both dividing and non-dividing cells.
So we focused on the work of Dalkara et al. For this study. They wanted to generate a virus using directed evolution, synthetic virology, that can penetrate deeper into the retina. And so they used 3 AAV2 libraries, which were mutated in 3 different ways. The first was with a random 7 amino acid sequence inserted into loop 4 of the viral capsid. The second was with a tyrosine to phenylalanine mutant, which was subjected to random mutagenesis. And the third was a mixture of chimeric capsids with random parts of AAVs 1 through 9, so the viruses all had mixed capsid structures. They injected the libraries into mice to see which were the most infectious, and then they took the most infectious of all these 3 libraries and mutated them further and tested them again, repeatedly.
So they found that the library with a random 7 amino acid insertion was most strongly preferred. If you look at panel A, you could see the 4 top clones that were used. 77% of them all ended in TRP, or threonine arginine proline, so they considered this as their consensus sequence. And in their future studies, they're going to use a clone called 7M8, which featured this specific sequence. Panel B shows the overlay of a typical AAV virus capsid with their 7M8 mutant, and the orange loop is the insertion that they made. And you can see in C and D that the loop 4 and loop 3 usually are located very close to each other, so they think that some interaction between these resulted in the new capsid characteristics. So moving forward, they took the 7M8 virus and a normal AAV as their negative control, and they used these to deliver GFP genes to the retina of non-human primates. And then following this injection, they imaged the cross-section of the retinas to test for the GFP fluorescence, and they found that 7M8 was very successful. You can see in panel E, which is their negative control, that there's very little fluorescence as opposed to F, which shows that the virus was able to successfully penetrate all the way through the layers of the retina.
Analyzing the results, the authors concluded that 7M8 is better than AAV2 when it comes to retinal transduction efficiency. Using 7M8 is also better because of its high infectivity, which can reduce the chance of immune response to the virus. The success of 7M8 also serves as a model for curing various forms of retinal degeneration in the future, and its engineering mechanism can be used as a source for other gene therapies where delivery across complex tissues is necessary. That is all, and thank you very much for watching.
I'm a virus, an infectious particle, an anathugger pirate, the genuine article, Ebola, chickenpox, West Nile, influenza, yellow fever, AIDS, herpes, SARS are coming right at you.